So I think it's a good opportunity to say, on the one hand, thanks for all the three speakers for great talks, but also to sympathize with the listeners. Maybe it's a good time to take a good breath, maybe to have a stretch. There's a good chance, at least that happens to me all the time, that even when the speakers do the best to explain, I feel saturated and overwhelmed. But at the same time, we want you to stay with us for a few more minutes, because we want to have both a discussion what we saw and to see if there's questions coming up. So if you feel like, it's overwhelming, it's like what most people feel. And there's a very known thing that happens to people in our profession. It's called the imposter syndrome, where people feel that actually all the others, all the hundreds of people listening to this really understand everything and follow everything, all the slides, and only they are kind of slow and dumb because they couldn't follow what was happening here. So I have a surprise for you. It's already has been researched, and it's been found that really the vast majority of people are feeling that, the same feeling that you're feeling right now, that this is too much for you. The other people are smarter than you are. It's very, very common. If you don't believe me, Google it under the imposter syndrome. Okay, and in that spirit, I think we want to challenge the panelists with either your questions or some broad view things for discussion. And we might also have some extra websites and resources that I think Janine is highlighting. And if you want to follow some of those websites, they're the most, the best things that our speakers thought that would be of most value. So I'd like to, okay, so everybody took the breath, had the time for a stretch. You can do it, in any way you want, because we don't really see you. So that really gives you even an advantage over all of us speakers. And I want to start with trying to think about, for each of the speakers in their field, something about the best case scenario as a community. So trying to think, be realistic, but still optimistic about, which environmental challenges you think we could tackle. If you have a quantitative bent to it, I think that helps, because usually things are on a quantitative spectrum. They're not just black and white. And if you could say something about where does life scientists, which is much of our audience, where do they have leverage and to which type of questions? So you can either jump in, or we'll go with the order of the speakers of Dianne and then Rob and then Toby. But if somebody wants to jump in, you could go ahead. I think you can all unmute yourself.
Okay. Dianne, you want to go ahead, or?
Sure, I'll just pick up where you left off, Toby, because I really loved your final sets of slides, and I thought it was wonderful how you highlighted that in addition to the carbon cycle, the nitrogen cycle and phosphorus cycle are very important, and of course there are other cycles too. But maybe that's a good place to begin. So I do think that it is important for us as a community to identify where the issues are that are actually tractable based upon the type of experiments and knowledge that we as life scientists can get. And I like the phosphorus and the nitrogen cycle and the carbon cycle very much, because I think all fit that bill. So as you pointed out, we're running out of phosphorus. When that will actually happen is not entirely clear. Some estimates I've seen on the order of a century. Certainly, it behooves us to be able to use phosphorus much more efficiently and sustainably. And I gave one example from my talk of how we might take a hint from microbial strategies that clearly are ones that potentially have something to offer, but that was just one example. There are many other examples where fungi, this is the reason I provided the link to Toby Keir's website, fungi are other microbes that don't get nearly enough airtime that are absolutely vital to the health of plants through intimate symbiotic associations where they help deliver not only phosphorus, but water. Thinking about water delivery, especially as we're going forward now with global change, there are going to be certain parts of the world that are going to become much more arid. Thinking about how we can tap into microbial communities to actually capture water more efficiently. I think these are topical areas that are actually very important. Numerically, Ron, I don't have a number off the top of my head that I can give you. I put that to you as a challenge. Maybe you can say more on that topical aspect, but I think actually I don't know if it's understood. I don't know if we know well enough, and I think that's something that we as a community need to think through a lot harder, which are what are the problems? What do we think are the potential solutions? And then do the calculations to ask, if we improved phosphorus recycling, for example, in a soil by X amount, what would that translate to in terms of ability to conserve phosphorus resource? I mean, you could do that kind of analysis on many topics. I'll stop and give the baton to someone else.
Right, maybe I jump in. Or Rob, I don't want to skip you. So my number in my head is 8 tons per CO2. I think we should all get used to think how much CO2 we are emitting, as much as we sometimes start to count calories in these days, right? So we need to get a healthy understanding what each individual is contributing to CO2 emissions. This is the first step to get to become more quantitative and also scientific in the ways in making decisions, right? For instance, that we meet now online is a very good decision, because I think we saved a lot of CO2. So I also think about other numbers, which are how much energy comes to this planet. And Dianne, you know this better than I do. Ron, you know it even better than Dianne probably. So it's 100 watts per square meters, at least in Germany, right? And we're just able to capture 1% of it. So in principle, it's enough energy there, right? But the question is, how can we become more efficient in harnessing this energy and using the energy on this planet to influence, in my case, of course, the carbon cycle? So can we, for instance, combine chemistry, photovoltaics, together with a biological system? Can we create this biohybrid system? Some people, especially also Ron, is working on. And can we leverage the power of biology in combination with great material sciences? And again, I think this is a vast untapped potential for us as life scientists to move into this area. And I'm just fascinated by the possibilities in there. And I think we should always be open to learn from nature, but also think how we can improve and or implement these natural principles in, let's say, more technological setup. And to give you one last example why I think it's important. So we all know how birds fly. We're all amazed by the flight of birds, and we've studied it for centuries. At the end, we build planes. They don't look like birds, but they mimic those principles that we've learned from the natural system. And maybe the CO2 fixation principle is not, let's say, photosynthesis, but the principle of CO2 fixation in a more technological setup. And I think we should not stop thinking about how to use those principles we learned from studying natural systems and to create new solutions. And this is, I think, what I would find most inspiring as a biologist these days. Okay, Rob.
Yeah, I mean, those two sort of comments are tough to follow. I would say something maybe more on sort of following on in terms of, why do we care about microbes so much? And I think the reason being is that microbes have basically exploited every single niche on this planet, and they are highly adaptable, and their ability to change due to their relatively short generation time is something that has made them so successful as a set of organisms. But as Toby mentioned, in terms of things like plastic, it's a really new substrate for these microbes to see. So there are what we're currently, the area where we're looking at are finding very sort of generalist enzymes. And then we have to then use the techniques, high throughput functional screening with mutagenesis to then kickstart that to try and get them to adapt. So you're accelerating that evolutionary process. We shouldn't be scared of doing that, but we also need to be just mindful of how we present that to a wider audience about what we're doing and what this represents. Everyone was probably familiar with the genetically modified crops and how society misunderstood to a certain extent what they were bringing, and that you can get a really negative pushback if we don't engage appropriately. So I think there is a little bit of, we've got huge potential there. I think the adaptability of microbes and the ability for us to genetically manipulate them is fantastic, but we just need to do this in a considered way. And I think there is also a notion of, we have to be mindful of the digital sequence and making sure that Nagoya is reflected. So this is the protocol to make sure that there is benefit sharing across the world. So companies going, companies going in, mining these data sets, they should not be allowed to just make a massive profit because, that sequence has come from an indigenous country where maybe they haven't got the capabilities to exploit it.
Okay, I think I got much more than I asked for. So we got perspectives both on where the life scientists, but also touching on some of the real challenges that we're facing as science wants to make an impact and acceptance, etc. I'd like to also have people touching on things they've seen in their field that is inspiring to them. It could be some study, it could be some website, it could be an initiative that they want to highlight. So now is your opportunity to do that.
I'll go first, sure. Someone needs to talk. I have given a couple of websites, both in my talk and have been provided. I encourage you all to look at them. These involve quantitation of human impacts on the biosphere, and also a wonderful website looking at the Society for the Protection of Underground Networks, talking about the importance of soil fungi. But as an example of research, to just get back to the point that the microbial world is really quite extraordinary as it is, and if we understand how things are limited, we can control them better. I'll point to research that was done in, I believe, 1993, in an experiment designed to try to understand why there were regions in the equatorial Pacific that were so-called high nutrient but low chlorophyll, where there was high nitrogen, high phosphorus, and yet there weren't very many phytoplankton growing. And the answer turned out to be that there was a limitation for iron. And so an experiment was done where a boat went over, like 60-something square kilometers and put experimentally into the oceans a certain amount of iron chloride. And sure enough, there was a resulting bloom in phytoplankton. Now, one can debate whether or not this is a good idea. One would need to know the unintended effects of something like this. But what I think it demonstrates, and the reason I bring it up, is that as we better understand what limits microbial processes that we care about, we can think about how to stimulate them. And so there's an example of something occurring on a large scale due to the insight that came from knowing that trace metals can limit microbial growth.
I'll pitch in with a different site. So I'll pitch in with the Tara Oceans project. So I passed the URL to Janine, and so I think this is a really good example, or the best example of where a global initiative of open data and really well-structured data has really sort of given us that ground zero of starting to understand the health of the world oceans. And I know there was a previous session on marine microbiomes, and that was probably covered to some extent there. So I would say that that's a really good example. If you want to start thinking about a multi-omics, multi-faceted project that has then continued and grown, then I would really recommend going and looking there. And they are tackling different issues like plastic pollution now in the world's oceans. And I want to make sure Toby's got enough time.
I mean, the great points you all mentioned, I can fully subscribe to your statements. I want to give a different angle to what both of you have said, and maybe also emphasize technological opportunities. When I did my PhD, I mean, we had to clone genes by hand, but how hard had it been? Now we just order genes, right? So also the high throughput capabilities, the microscope you can use, the metagenomic, genomics, proteomics, machine learning, big data, and analytic capabilities we have, speed up the discovery and also design of our experiments. And I think being more quantitative about this is very important. being also a bit more synthetic is very important because it allows you to test hypotheses. And I ask myself also, how does the future of lab look like in 10 or 20 years for us, or for you as trainees right now, right? So we can leverage the power of living systems by manipulating them really hard. And Ron has done great examples of showcasing this and learning again back what happens if you challenge those systems, which tells about the flexibility and how fast things can adapt. And this is something which is pretty hard to predict these days when we change, let's say, the global carbon cycle so fast. I mean, there is a natural limit how fast organisms can adapt to that. Microbes might be the best in it. Rob pointed it out. They've been around for 3.5 billion years, but we also know this is probably not us human beings being adapting so fast. So I'm just amazed by those works that also try to do experimental evolution. And what came out of Ron's lab was really fascinating for me. And I think this is probably maybe a good ending and handing it back over to Ron.
So I see also in the questions that we're getting a lot of emphasis on evolution and how can we consider doing something that's that would be, on the one hand, leveraging from evolution, and in that respect, there's the challenge that many of the microbes that we might want to leverage are actually, there's a question of whether we could culture them in the lab or not. So maybe on those challenges, anybody wants to reflect. And at the same time, maybe another related question is about how could we, given the 3 billion or 4 billion years head start, how can we think that we might be doing things differently, better? What is really happening here? So maybe touching on that question from the audience.
I would love to jump in on that because I think that oftentimes there's a statistic floated around that only 1% of microbes in nature are able to be cultured. And I want to demystify this because I think that that's a number that has been overplayed and may not be accurate, even when it was first put out. And it certainly isn't accurate now because we're living in an era where there has been tremendous acceleration in our ability to understand the environmental chemistry in which interesting microbes reside. We have a much better appreciation for their interactions with each other. Technologies have advanced so that we're able to cultivate organisms at the single cell level now, allowing slower growing bacteria or other types of microbes to be separated and brought into isolation. In addition, there have been unbelievable advances in genetic technology so that now it's extremely easy to be able to manipulate just about any organism. So these things together provide just unbelievable opportunities to go into nature and to enrich and select for catalytic abilities in the microbial world that are of interest, which is sort of the first step towards being able to later do synthetic biology and engineer and adapt them, as Toby explained. So I think it's an exciting time to be a young person in this field. And I'll end there and see what others say.
Maybe I should demystify our picture of evolution as being the most creative force on this planet. I fully agree, right? But you should not forget how evolution works. It always builds on existing parts, on existing subparts and existing systems, right? And so in the beginning, it might be really rapidly scanning, let's say, the space until homing in on one certain solution. Then it will evolve and improve the certain solution, finding a local optimum, but not maybe the global optimum. It's us as human beings being very progressive as teenagers and becoming very conservative at the end. So, and being able to replay the whole system and setting it to zero and seeing how fast it could evolve, this is fascinating to me. So what I want to say is, there's this strong historical dependency. If you evolve a new metabolite in the context of a metabolic network, you want to make sure that it doesn't disturb 3,000 other enzymes in the context of E. coli cells or the genetic network. So there's a lot of constraints. Evolvable a system is once it has reached a state, like after 3.5 billion years of evolution. There's Windows 11, Windows 13, Windows 14, but maybe it's better to have an iOS system being built or Linux at the end of the day to restart new possibilities. And I think this is what we also should appreciate as biologists, that evolution, yes, is creative, but it's also maybe homing in and evolving in one direction. It's pretty hard to turn it back around. It's a one-way road to some extent due to dependencies that exist. And David Baker has shown that. David Baker has shown there's many more protein folds that could exist in theory, but were never used by biology. It always builds on, let's say, 10 or 20 different folds and reshuffles those folds, but doesn't move into this other space for what reason ever. So don't overestimate the power of evolution. That's my 2 cents.
So I'm going to come back to Dianne and just on her point. I agree that the way that things have yet. So I always refer to it as yet to be cultured, not to say they're unculturable. That's always my way. And what I would say is actually that we've already started having some success in looking at some of these poorly represented clades and actually using just the metagenomic information to actually guide the culturing techniques to actually work out what is this microbe deficient in terms of pathways to then say it should have this. I think the notion of actually going across, going away from monocultures and having simple communities is going to be the next big wave in culturomics. But I would just refer back to the my protein database in terms of when we talk about sort of 2.4 billion sequences. It depends if your glass is half full or half empty. I'm going to be the pessimistic person today. So only half of those we can actually put any form of functional annotation on, which means the other half we have no idea. They're just a sequence. And I think that to me is the challenge. And Toby, you mentioned it as well, in the fact that, what are all of these doing? Now, we are beginning to understand a little bit about what these are doing. And I think there are a lot of those things that don't get labels of just some of that is bias about how we label things. I think there are still some really interesting pockets out there of things that are either very different to what we know or are completely new. And I think that is both a challenge and an exciting area that I would want to go into if I was a bit younger.
Fantastic. I would suggest that you maybe consider it, no matter what's your age, to take on another challenge. I think we will soon be moving into the opportunity that we have for the audience to meet the speakers. So we'll take just a few more minutes and then we'll move into the next stage in which you can really get to know people in a different mode rather than just this one. There was a question that was geared to Toby about whether we could expect nature and evolution to have found other pathways for CO2 fixation that might be found in the near future. Do you want to mention something about that?
Sure. So I want to start with that. Let's say genomic and metagenomic evolution has allowed us to look and find bugs that can grow with CO2, a source of carbon, but apparently don't use the Calvin cycle, which has led to the discovery of 3 or 4 new CO2 fixing pathways. I also want to mention that, let's say 2-3 years ago, a couple of years ago, we also had synthetic biologists like Aaron Bar-Evan thinking about pathways like the reductive glycine cleavage pathway, which was designed first on the paper and then found later as being actually existing in nature. So it tells you already that some of the things we think of, maybe the catch cycle, could actually exist also in natural systems. But knowing how these systems look like, we can now harness and go through metagenomes and look which bug might have all the key enzymes required. And that's of course exciting for us too, to see if there's even more pathways out there. It's hard to find them, but we can find them. And I think we've seen over the last 10 years, 3, 4, 5 new pathways being discovered. The question of how big is the niche you're living in, right? I think that is of course defining also the success of the discovery. Yeah.
So thank you for the Siva Sanka from the audience for bringing up this question. And another question that was raised was in relation, maybe it's relevant for all 3 speakers, about the fusing of enzymes. So Orakov Askalbek is asking about whether fusing of enzymes could improve pathways and efficiency. And I think that might relate to each of the speakers today.
I can go. So fusing of enzymes to complete a pathway presents challenges. So usually within a pathway, there's one rate limiting step. And so if you're constantly fusing things together, you've then got a pair and you're carrying around something extra that you don't necessarily need to do. Often making a fusion also limits the dynamics of your substrates flapping around. It can also change things like that. So I think that, you know, there are different techniques that I hear about in terms of actually having really smart systems of actually how you can cascade substrates from one as they're getting processed. And I'm not the person to answer that. I think I'll hand the sort of more wet lab answers to my esteemed colleagues.
I'll just take this as an opportunity to point out that classical genetics and biochemistry really never go out of style, and that if we want to fundamentally understand a catalytic process, we need to do the hard work to figure it out piece by piece, because the answer is going to vary depending upon the pathway that you're talking about. And so it's very difficult to generalize, and it's important to be precise. But this is where my optimism returns, is that I think that nowadays there's an opportunity to do this type of mechanistic research with a glorious diversity of microbes if we're selecting for phenotypes that we care about. Where we have a phenotype, there is now a path to figuring out how it works. It requires a commitment, but it's an incredibly exciting opportunity for fundamental discovery.
I would love to mention and to thank back the person from the audience. The name is Vesta Patrasionate, who was mentioning the big thank you for the organizers. So I'm not one of the organizers, and therefore I could thank them also on everybody's behalf. So it's a really collaboration between the HHMI, the Howard Hughes Medical Institute, and the EMBL, the European Molecular Biology Laboratory, which I think, you know, if you're interested, everything that you saw now, if you want to share with everybody, that's been recorded and will be available on the website. But also, this is a part of a series of lectures with institutions, leading scientists from around the world on varied subjects. So you could share also that with many other people that I think might find interest in that. And that's one of the beautiful things in the world today, how it's so, you know, not only is it not only cheap, it's all free, it's available, all of that knowledge, insights, etc., to everyone. So thanks to the organizers, and we'll now take the next few minutes to have the opportunity for people to interact directly with the speakers. So we'll be led into the Meet the Speakers session. Thanks everybody, have a wonderful day, and choose good problems and get them done.
Wonderful. Thank you guys so much. Thank you to you, Ron, and of course to Dianne, Rob, and Toby for a really fascinating session today. Thanks to our whole audience for joining us. As always, I do want to mention that all of the websites and resources mentioned by the speakers in their talks are noted in the document that I shared in the chat box, and they'll also be available on the Life Science Across the Globe website, along with the recordings of today's session. You know, if you would be so kind, we would love if you would take a moment to complete the survey that I linked in the chat box. And also don't forget to join our next Life Science Across the Globe event, which will take place on June 1st at the same time, 9 a.m. Eastern, focused on air quality and infectious disease, hosted by the Institute of Infectious Disease and Molecular Medicine at the University of Cape Town in South Africa. So we're really looking forward to that.